I'm going to initiate the seminar. I'm introducing Kartik Chandran today. I'll give you just a tiny bit of background. Kartik is a superstar virologist at Einstein. He received his bachelor's degree from Lafayette College in Pennsylvania and his Ph.D. from the University of Wisconsin. His mentor in Wisconsin was Max Nibert, a well-known virologist. Kartik pursued research at that time, I believe, mostly on reoviruses. He continued as a postdoctoral fellow with Nibert, who then relocated to Harvard, and Kartik followed him there. And then shortly afterwards, he transitioned to another lab at Brigham and Women's Hospital in Harvard, which was run by James Cunningham. Kartik entered into work on Ebola and other filoviruses during that period. He made some truly astounding discoveries about the entry pathway of Ebola viruses, which he has presented to us a number of times here at Einstein in faculty seminars. And he obviously won't be talking about that work today, but we'll be discussing his current efforts to contribute to our understanding and development of therapeutic approaches for the novel coronavirus. I can mention also that on our faculty, Kartik has been an absolutely outstanding, successful member of the M&I faculty from the moment he joined us in 2007. He rose to associate professor on target in 2012, and then had an accelerated promotion to full professor after 4 years in the associate rank in 2017, and received tenure in 2018. He has a expanding laboratory that is extremely dynamic. You can see, I think his slide is showing now, you can see, I don't believe that's his entire lab, but that's most of his lab, which has continued to work very hard throughout this pandemic, even during the lockdown period when most of the people, or all of them that you see on that slide, were designated as essential personnel and continued to work really pretty much around the clock. I don't believe I ever saw the lights off in Kartik's laboratory anytime in the last 3 months. I don't know exactly what Kartik's going to tell us today about coronavirus, but I know it's going to be interesting, and I know it's going to be important just based on the work that he was doing prior to the pandemic on Ebola and other filoviruses and hantaviruses. So it was a kind of a natural, easy step for him to take to move a lot of his expertise to the coronavirus problem. I will now turn this over to Kartik for his seminar. Thank you.
Hi, good afternoon, everybody. Can you hear me okay?
Yeah, you're fine.
Great. Thank you, Steve, for the intro. Thank you, Aviv Bergman, Dean Tomaselli, for the opportunity to share with you, with all of campus today, some of the work that we've been doing on COVID-19 since the pandemic started. I'm going to try to tell you, a few different stories about the work that we've been doing. All of them are still in progress, and I think you'll see that. But I'm also really looking forward to your input and ideas on how we can move this forward, and also, through the collaboratories as well as other mechanisms, how we can synergize with other folks on campus to ask questions that we're not even thinking about right now. A lot of the work I'm going to show you that was done in my lab was done by this group of folks that we're informally calling the COVID crew, and Gorka, who's a computational biologist, Gorka Lasso, in my lab, who also assisted in some of these efforts from working from home before phase 1 started. And I'll call out people as we go. But I also want to point out that the work I'm going to tell you about today is part of a much larger collaborative effort on campus, as well as with other collaborators off campus, including most notably John Lai's group, Lizan Pravsky, Johanna Daly, Steve Amo, We have folks in the pathology department, Michael Prstowski, Amy Fox, Yitz Goldstein, the biorepository at Einstein led by Dan Stein. So, it's really a very sort of, I think our goal was to just pitch in and do what we could to help to apply our specific expertise to modify it to study coronaviruses and to do things in the clinical realm that we don't have a lot of experience with. So it's been a learning experience. It's been an amazing, sort of challenging and amazing journey working with folks all over campus and at Montefiore. And I will call out as we go as best as I can all of the different folks that were involved in the various projects. So I'm going to start off by telling you about something that is, we're just getting into gear now, but something that we think is a very important question, which is to actually sequence viral isolates of this virus in the Bronx. And our motivation really was, our goal to do this, even though we actually had no prior experience in viral sequencing, it was driven by the fact that New York City has quote unquote acted as a grand central station for this virus. This is from an article today in the New York Times. The Bronx in particular has the city's highest rates of diagnosis, hospitalizations, and deaths. However, New York City sequencing has focused mainly on Manhattan cases. There are very few Bronx sequences available, something that John Greeley has eloquently spoken about before and has really advocated strongly for us to work together to try and get more Bronx sequences to answer the question, what does viral diversity look like in the Bronx? How can we relate viral genotype to patient outcomes? Can we say anything about that relationship? And can we make any inferences about the spread of this virus in the local community? And this is one of a number of different collaborations that are overlapping that have driven these projects. So in order to do this sequencing, to actually do the analysis, we're working closely with Libusha Kelly and also Aviv Bergman, John Greeley. The samples have come through our collaboration with Yitz Goldstein and Amy Fox in pathology, who've gotten us remnant swab samples from hospital patients here. Since we didn't have a lot of experience doing viral sequencing, we decided to jump onto a platform that's sort of been crowdsourced and made available for everybody that's trying to sequence. It relies on the MinION, which is a nanopore sequencer that was actually, to my surprise, relatively simple to set up. And the workflow for this is to isolate viral RNA from these remnant nasopharyngeal swabs from patients that are PCR positive to carry out a cDNA synthesis reaction and then a tiled multiplex PCR to generate a library that is barcoded and then can be pooled to look at multiple sequences at once. This is then run on the nanopore MinION sequencer, and then base calling analysis. There's a whole analysis pipeline of base calling and phylogenetic analysis, and then we're just getting rolling here. So even just before this call, Aviv and I were chatting on the Zoom, and there's a lot of different things that we can really do here to leverage, all of the expertise in the Einstein community to mine these sequences for as much information as we can get. And so just to sort of point out some of the folks. So Max Fels, who recently graduated from my lab with a PhD and is staying on to do a short postdoc, he was really the one that drove this. He came to me one day and said, I really want to do this because there are very few sequences. And so I was like, let's do it. And that's kind of how it all got started. Karen Scalina in pathology, working with Yitz, has helped us get these samples and to curate them and to also obtain clinical information associated with these samples. Saad Khan and Ryan Forster in Libusha Kelly's lab have been working with Max closely to actually build this pipeline to actually analyze the data, to do the base calling and the variant calling, and then to do the phylogenetic clustering and the analysis. And this is something, we have a Slack group together, and there's a lot of activity on this Slack group as we're trying to figure out, how to actually build this pipeline, how to get good data, how to call variants that are reliable. And so it's been a very interesting experience and continues to promise to be a very nice interaction that's going to teach us a lot about these sequences. So just some very preliminary data from a handful of sequences that we've assembled so far. These are full genomes, by the way. What we find is when we look at some of these trees is that Bronx isolates are more closely related to New York and Connecticut cases, as opposed to these outgroups that are Wuhan, Australia, Midwest, Brazil. Not so surprising, and sort of a sanity check, I think, just in terms of getting going with our studies. Interestingly, we find that all Montefiore isolates that we've sequenced so far have this particular amino acid change in the spike protein, D614 to G, which we don't really completely know what it's doing, or if it's in fact doing anything, or is just the result of a bottleneck event. But certainly almost all the cases, all of the viruses that have been sequenced on the East Coast have this mutation. And there's some recent evidence that it may confer some sort of selective growth advantage on the virus, but the jury is very much out on this. But it is interesting that all of our sequences have it. And it's also interesting that all of the sequences that we've gotten so far clustered together with other Bronx isolates. So we're sort of just setting the table here for getting a lot more sequences now that the workflow, both on the wet bench side as well as the analysis side, has really been set up. And we're hoping to really build a much larger database of sequences that will be made publicly available and that we can mine for more and more information. I look forward to working with Babusha, Aviv, John, and others at Einstein to really try to use this resource to the best of our ability. So that's sort of where we are with the sequencing. I'm sort of trying to start from detecting the virus, sequencing it, to actually figuring out what makes it tick, and then I'll sort of close out with what we're doing in terms of developing therapeutics. So I hope there's a logic to the progression of the talk that you'll appreciate. So I'm going to move on next and tell you a little bit about the project we actually started first when the outbreak really started to ramp up in early March. And that was following a conversation with Dr. Lizan Porofsky, who was driving as a PI of a proposed RCT, or randomized control trial of convalescent plasma as a therapeutic. And Lizan and Johanna Daly had already been collecting recruiting donors to collect sera and then hopefully plasma that could be used in a therapeutic context. And Lizan reached out to see if we would be able to do some serology on these samples to actually determine the nature and measure the antibody responses in these samples from COVID-19 patients and convalescents. We, of course, we'd set up assays like this before, but hadn't really worked with clinical samples. So this was sort of a new project, but this seemed like a way for us to jump in and pitch in and do something helpful. So we got on board. And so then the question was, can we identify convalescent plasma or CP donors with high antibody titers that could be used to treat COVID-19 patients? And what high titers means we can sort of unpack as we go along, which I don't think we still completely understand what this means, but we certainly are trying to look at as many different variables as we can. We want to also understand what are the correlates of protection afforded by convalescent plasma. This larger project is seeking to understand that. And then we want to know if we can mine B cells from convalescent donors, or maybe even acutely ill individuals, to identify potent monoclonal antibodies that could be used for prophylaxis and therapy. And I'll try to get into all of these different aspects of this particular project. So in order to set up an assay that would allow us to do something very simple, which is to measure the levels of IgG against the SARS-CoV2 Spike protein, we needed an assay to do that. And that assay and this larger project was born of a collaboration with Lizan, Johanna Daly, and also John Lai in biochemistry, who's a close collaborator of ours over a long period of time, as well as Steve Almo. And so I'll tell you more about how what everyone's doing there. But the plan for recruiting potential donors was as follows: is to advertise a study to find the subjects, survey to pre-qualify healthy volunteers, and then invite them into the study where a small sample would be drawn for preliminary analysis. And in some cases, we also collected peripheral blood mononuclear cell, or PBMCs, for analysis and for use in antibody discovery. We then measured antibody titers to the SARS-CoV-2 Spike, as I'm going to show you. And if folks met a certain cutoff, then we referred them to the New York Blood Center for convalescent plasma donation. So the criteria initially, at least for recruiting these subjects, was a positive qPCR test, followed by course of illness, and usually in this cohort, it was quite mild. These people have been well for at least 2 weeks, and they were healthy. So Johanna and her team were able to survey over 1,500 healthy volunteers, and recruit 294 to this initial stage of sampling to determine their sort of serology. And then of these 294, approximately 190 were referred to convalescent plasma to New York Blood Center for convalescent plasma donation. I don't actually remember off the top of my head how many people actually went, but if you know, we can ask Johanna afterwards maybe. And we also collected PBMC from 33 different volunteers for analysis. And we're actually continuing to do longitudinal follow-up on some of these individuals. So where did Johanna go to find these donors? Well, one of the first big clusters happened right just barely north of us in the New Rochelle area. And so Johanna collected at New Rochelle and Scarsdale and Muncie, but she also collected from Montefiore as well. And that's sort of the makeup of our donor pool. And these are just some pictures of all of the folks, including Johanna, that really went out every day to collect these red tops. And for a while, you know, the only thing you could hear around the lab was red tops coming, red tops coming. So that was, you know, there was a huge rush to try to get these donors and very quickly do their serology to try and recruit them for this convalescent plasma studies. So some of the characteristics of this cohort, these are just some of the data. Again, the key point here is that these individuals had gotten sick and most of them had a confirmed swab, and they had since recovered and had been symptom-free for at least 2 weeks. So our first task in the lab was to develop a serologic assay for spike-specific antibodies. And just to orient you a little bit, this is actually a very cool sort of image of a model of a coronavirus particle by David Goodsell at Scripps. And so this is the purple sort of ring you see here is the viral spike glycoprotein that forms these trimers. That it's a membrane-bound protein that's sort of embedded in the viral envelope that's shown in gray here. And the spike protein is the major viral surface protein of all of these viruses, including SARS-CoV-2, and is the major target of antiviral antibodies. So the first challenge was to actually produce an antigen based on the spike protein that could be used for a serologic assay. Our plan was just to do something quite simple, like an ELISA that could be set up easily in the lab, and is also the sort of assay that can be run in parallel at different sites. But it relied on this antigen being available that mimics the authentic spike trimers on the particles, but can be produced as a soluble protein in large amounts. So the original construct design for this was done by Jason McClellan's group at University of Texas, Austin. Jason is a close collaborator of mine and John Lai's as part of this larger U19 project called Prometheus that I'll tell you more about. And so Jason generously shared this plasmid with us. But Jason is a structural biologist and as well as a vaccinologist, and his goal wasn't necessarily to produce insane amounts of this protein for ELISA assays. And so, however, John Lai and Steve Almo's labs took up the challenge to actually do the biochemistry to actually produce large amounts of a very well-behaved protein. And they were able to really produce many milligrams of this protein in a suspension CHO-based cell line, which has really facilitated and driven this work, and also allowed this assay to be able to be used by the Monty pathology department in the clinical lab as an actual clinical assay well into the future. So this sort of, allows us to run this assay even if commercial vendors aren't able or like out of stock of the reagents or what have you, because this is really built in-house using reagents that can be easily purchased for research, as well as this critical spike protein that's being produced here at Einstein. And what I'm not going to show you, and I think Steve will probably talk about, or John, is the characterization of this protein, including its structural characterization, to really show that it makes these beautiful, well-behaved trimers. And this is what we used to set up a very simple ELISA-based assay where we just coat ELISA plates with this recombinant spike antigen, and then come in with control antibodies or with these different convalescent sera from these different donors that were collected. And then you can sort of see, I'm just showing you like sort of a sampling of what these ELISA curves look like. And you can sort of see that there's they span a gamut of reactivity. These individuals down here with very low reactivity and these two individuals, 28 and 43, that have much, much higher signal. So we can use these full ELISA curves to estimate an ELISA titer, and that is the number that we're really using in the context of the convalescent plasma studies and the trials to determine a cutoff, which I'll tell you a little bit more about. So one thing we really like about this assay is it's very robust, it's very reliable. The data I'm going to show you were actually run in our lab, in Johanna Daly, in John Lai's lab, and Lisa Amprowski's lab by different people. We set up a system where we essentially, our lab became sort of a reagent distribution hub where people could come and they would have samples to run and they would get all the reagents. So, and then we collected all of the data into one place, standardized everything, and did all the analysis. So it was really a distributed operation to do serology on these hundreds of samples that was done in multiple labs. And each sample was run in parallel independently by 2 different people. So we could really rely on the data. And so, I mean, this was really an amazing effort by all of these different folks, not just in my lab, but also in the other labs that I mentioned. So this is just sort of a summary of that data that we obtained. What you're looking at here is, on the y-axis is a titer on a log 3 that's been converted to log 3. And the reason why it's a base 3 log is because we did 3-fold serial dilutions of the sera, so it was just easier to get these round numbers. And so you can see that there's quite a spread, but the median titer is about 7,000, which is very respectable. Just to kind of give you some perspective, and every ELISA assay, unfortunately, every one of these serologic assays is different and uses a different cutoff or a metric, which is a little unfortunate, but this number seems to be much higher than the sort of titers that Mount Sinai, for example, use as their cutoff for convalescent plasma donor recruitment. So it's a healthy, significant, robust amount of antibody. And fortunately, this is the median, and we're able to find lots and lots of people that are close to or above it. But we set a much more conservative threshold, which is a titer of about 1 to 1,000. And that's shown here by the blue line. And we only referred folks above this threshold for plasma donation. And this is similar data, but obtained now from plasma segments. So these are these little tails on the plasma bags from when people have actually given plasma. And what was really reassuring, and we were really worried about this at first before we did this, but it was really reassuring to see that the donors have very similar titers in terms of distribution and now here we're looking in plasma that in the bags rather than in these little red top serum samples that were originally collected several weeks before. And you can see that these people have similar titers, and we also have lost this tail down at the low end, presumably because that's the cutoff we set. So this is very reassuring that these are the plasma bags that are going into individuals receiving convalescent plasma therapy. So another question we asked is, do these donor plasmas contain neutralizing antibodies? And so I'm going to tell you a little bit more about this in a little while. In order to very rapidly screen for the neutralization activity, which is the ability of antibodies to block viral entry into cells, we had to set up a high throughput biosafety level 2 assay. This virus is a biosafety level 3 virus. We don't actually have a virus biosafety level 3 at Einstein, although we're working on that. So we needed a surrogate assay that could very quickly give us this type of answer. And we've set one up successfully, which I'll share some of the details a little bit later. But using this assay, we measured the ability of these sera to block infection at a specific dilution, about 1 to 100. And then we actually correlated the ability to block infection to the titer across this whole sample set of more than 250 samples that we've collected. So on the y-axis, you're looking at infectivity. So 100 means no neutralization, 0 means 100% neutralization. So the lower the curve on the y-axis, the better the neutralization. And on the x-axis, the further out you are to the right, the higher the titer, the IgGs against the spike in these sera. So what you can see is that there's actually a pretty good correlation between the ELISA titer and the neutralization. So the higher the spike titer for IgG, the more potent the serum is at neutralization, and vice versa. However, it's also true that there are many exceptions. So especially when you look at intermediate titer samples, sera, where these are around the median in terms of overall IgG titer, but there's a huge spread in terms of neutralization, all the way from nothing to 100% neutralization at this specific dilution of serum. So this is something we're really intrigued about and really want to try and understand. What is it about these sera? What is it about the antibody composition that's different in these different sera that gives them similar spike reactivities, but different neutralization potential. And could this have some sort of clinical relevance in the context of individuals mounting their own immune response to infection? Could this have relevance in the context of convalescent plasma provision as a therapeutic or a prophylactic? We have some hypotheses, as I'm sure you do as well, and I'd be happy to discuss. So on this project, our ongoing and future work is to continue to do not just the donor sample serology, but also serology from the patients receiving plasma in moderately ill Montefiore patient cohort that has received convalescent plasma under compassionate use. That study is now ended and has transitioned to a randomized control trial, but we're still working through those samples and also working with all of these different folks and others now mentioned to correlate our serology and other sort of markers with patient outcomes. Similar studies ongoing for this RCT of convalescent plasma. That's a joint project between Montefiore and NYU. We're also, something I don't have a lot of time to talk about, we're also doing longitudinal serology on acutely ill and convalescent patient cohorts in collaboration with Amy Fox and Erica Orner in the Department of Pathology. And we're also starting to think about and do a deeper mining of the patient samples. So we're starting to look at other antibody isotypes, to look at other antibodies to other antigens in the virus, B and T cell responses, not just the humoral, like the antibody, the sort of the serum antibodyome, so to speak. And also what I alluded to, the composition of spike-specific antibodies with respect to epitope, neutralizing potential, and Fc effector function. So, stay tuned. Hopefully we'll have an opportunity to share, either I or one of our collaborators will have an opportunity to share some of these data with you. Okay, so I'm going to move on to the next part of my talk, which is trying to take this spike ELISA, which is really set up as a research assay to look at convalescent plasma, to develop a diagnostic antibody test for this virus. And why is this important? Why do we need this? Considering that there are many commercial tests now available and also increasingly coming online. Well, when we first started this, there was an acute need for such antibody testing. It wasn't entirely clear what were the good tests and which were reliable and which weren't, what kind of test. It was also a shifting landscape of commercial tests. That continues to be the case. Also, other things are stabilizing there. The sensitivity and specificity of these tests is not always clear. A lot of times samples are tested on relatively small cohorts that seem to be sort of handpicked. So there's a question about what happens when you look at lots and lots of people with varying degrees of disease or varying times post-infection convalescence. From a more sort of practical standpoint, it's also challenging to future-proof, doing antibody testing given the high demand and limited supply. And we really don't know what's going to happen here with the supply chain going forward. And so we really wanted to know if we could leverage the spike-based research ELISA to develop a clinical test. And so this is, again, a collaboration with many moving parts, many different individuals pictured here at Einstein and Montefiore that really helped us build this, take this assay from a research ELISA, validate it as a clinical assay, and then finally, hopefully it'll be final, but we'll see, submit a emergency use authorization application to New York State. So this assay, that application is currently pending, but it can currently, and then also, of course, upon approval, allow this clinically modified ported assay to be run in the Montefiore clinical lab. So I'm going to spend the next few slides telling you a little bit about this. So again, the issue here is having the spike antigen and having it at scale. And here as well, Steve Almo and John Lai's groups have really done incredibly important work of producing huge amounts of this spike antigen that's really well characterized and can be stockpiled to future-proof this clinical assay. So the data I showed you before with the ELISA was really done with these curves. So that's not really that helpful in a diagnostic test. We really want to turn these ELISA curves into a single cutoff value for diagnosis. And so essentially what we did initially was scout at 3 ranges of dilutions, 1 to 100, 1 to 1,000, and 1 to 10,000, to try to see which gave us the most sort of sensitivity and versus specificity, and also the most sort of dynamic range in seeing differences in levels of antibodies among different patients. And that's just the same samples rerun, but just those 3 different dilutions, showing you that, again, we can kind of pretty much recapitulate the sort of data we're getting with the full serial dilution curves. We did something similar with IgA, serum IgA, using a specific antibody to detect serum IgA. The overall levels are somewhat lower with serum IgA, which is not surprising, especially in these convalescents. But again, so we just picked more, and it's also probably the nature of the secondary. Regardless, we picked 3 dilutions here as well for scouting. Using this sort of information, we were at these 3 different dilutions and running lots and lots of samples. We were able to use data from single point screening of this convalescent cohort that I showed you data for before. This is this 224 convalescents, a December, January small control cohort that may or may not include some COVID samples for all we know. And then a much larger number of pre-COVID samples. And what this allowed us to do is really set a cutoff or a threshold for calling positive versus negative. We decided early on that we wanted a binary assay that was either positive or negative. We decided not to call it borderline. I guess this is something that could be revisited over time as assays run clinically. But at least at the moment, the assay is set up with this particular cutoff of absorbance at a specific dilution of serum or plasma. And it pretty well discriminates from positives and negatives, although you can see that some convalescents do actually have pretty low levels of antibody that are barely detectable and fall below the cutoff for being called positive. This is sort of an issue with a lot of the different diagnostic assays that are out there at present. We see similar results with the IgA. This should actually be IgA and not IgG. And so you can sort of see again that we found a different cutoff for the IgA assay that called most of the convalescents as positive and most of the controls as negative. However, you can see here that the IgA assay seems less sensitive in that many people that are IgG positive or fall below the cutoff for IgA. But we think that using IgG and IgA together may have some utility for identifying individuals that may be missed by either assay. This is just a summary of the results that we got. I'm not going to walk you through it. I'm just mainly going to highlight the sensitivity of this assay is in the high 80s. The specificity is in the 97 to 98% range. The specificity sensitivity is somewhat lower for the IgA, similar specificity. And we think that by essentially going from IgG to IgA, we'll have to see how this works out, but we think we'll be able to identify more positives. We certainly can in this convalescent cohort and bump up our percent positive by doing IgG plus IgA. We then also wanted to really look at the longitudinal sort of performance of this assay in terms of individuals that were hospitalized. So can we essentially identify individuals that are admitted to the hospital that are early in infection and should have low levels of antibodies? And can we see these antibodies go high over time? And so this is just data that I'm showing you here for a small group, but I'm just going to turn off my air conditioner. This is just a small data set I'm showing with about 20 odd samples that were early and late. So these are individuals that were hospitalized. There was blood draw done early and then a blood draw done about 7 to 10 days after hospital admission. And so you can see with IgG that many of these samples, although some of them start out very high and stay high, and this is something that we're seeing in this hospitalized cohort, which is very, very high spike-specific antibody levels. But a number of them do start low and go high. And this is just sort of retrospectively looking at days post symptoms based on the day of admission and self-reported information about how many days the individuals had been sick. So take this with this, somewhat with a grain of salt, but nevertheless, it does provide a longitudinal picture where you can sort of see that IgG can be reliably detected 7 to 9 days post symptoms. And almost all patients develop serologic evidence of infection. And we are actually now doing true longitudinal studies on more than 60 or 70 of these hospital cohort patients. Again, we're doing this in collaboration with John Lai's group and are doing this using a robotic setup that Olivier Vernoli in his lab has optimized and is running along with several other folks. So we will have some true longitudinal data that maybe John will be able to share or one of us will be able to share down the road. We see similar results with the IgA as well. We don't necessarily see the IgA is more sensitive for catching early cases, but it does seem to have a similar sort of performance characteristic to the IgG in these longitudinal studies. We also wanted to ask if our assay could distinguish SARS-CoV-2 from other sort of common human coronaviruses that are associated with infections. And so what I'm going to show you are some data with multiple ELISA. So what you're looking at here in this upper left quadrant is ELISAs with the SARS-2 spike antigen, the ELISA that I've been showing you data for. And you can sort of see here that So these are some samples that are selected because there was evidence from nasopharyngeal swabs of coronavirus infection. These samples were blinded to us initially in terms of what virus, but you can see that they all seem negative for SARS-2, except for this one individual and the control that are confirmed COVID. But these all turn out to be confirmed human coronavirus by swab, and they're all negative with the SARS-2. ELISA, we weren't sure about that one. But what's interesting is when you take these same patient samples and you look at other spike antigens from other coronaviruses, so 229E, MERS, and HKU1. So HKU1 is a beta coronavirus and is the most closely related to SARS-2. And so you can see that a lot of these samples that are negative for SARS-2 are actually positive for some combination of the other non-COVID coronaviruses. Not MERS, not surprisingly, because MERS is not that common in the US, but certainly 229E and HKU1R, and you can sort of see that this individual here had a 229E infection. We also observed that individuals that are very strongly positive for SARS-2 antigen can be cross-reactive for HKU1. That's not so surprising. But what we didn't see, and we have a lot more samples, is we don't see that HKU1 sort of confirmed patients are SARS-2 positive. So that, at least in that direction, we seem to be able to pretty specifically call a SARS-2 infection versus another coronavirus. This is similar data from a larger cohort from our Swedish collaborator, Matthias Forssell, at the University of Umeå in northern Sweden. They have a larger cohort of these pre-COVID swab-confirmed human coronavirus cases. And so you can see again that a lot of these, although not all of them, but many of them are positive by serology against antigen for HKU1 and 229E, and all of them are negative for SARS-2. So again, this is sort of true for both IgG and IgA. This just reinforces the point that we can specifically differentiate SARS-2 from other coronaviruses using this type of spike ELISA. So where do things stand with the diagnostic assay? We validated against Wadsworth New York State test, which is a totally different technology. We validated against Columbia's own laboratory-developed test, which is an ELISA-based test like ours. Our test is being run as part of a larger cross-validation study at Monty. That's almost done. As I mentioned, there's an application for New York State emergency use authorization that's in, and it's under review. The assay has been ported to automated to a robotic format at MM's Pathology in their CLIA certified lab and cross-validated by Sean Campbell and Jim Fay. And the deployment is expected any day now. So so far things look good, especially for the IgG. The IgA is still a bit of a work in progress, but we are hopeful and optimistic that that this assay will afford much larger throughput for this diagnostic serology to be done at Montefiore using this home-built assay. Okay, so I'm going to tell you a little bit now about some of the assays I've actually been talking about, but not really going into very much detail. And in particular, I'm talking about a BSL-2 infection assay that's needed to study viral infection mechanisms, screen small molecule inhibitors for antiviral activity, to profile the immunologic activity of candidate vaccines. I mean, there are literally hundreds of vaccines that are, in some stage of, discovery or development. And, even if you sort of consider just the major players, there's going to be a huge amount of immunological profiling of vaccinee samples, for example, that's going to need to be done. First, maybe in animals, but also obviously in humans. And so it's almost impossible to imagine that all of this can be done in a BSL-3 lab, given the limited number of labs there are, given the limited throughput that is really those types of assays can afford. Like a classic plaque reduction neutralization test, which actually looks at plaque reduction by doing plaque assays. I mean, that's just not something that you can very quickly turn around to look at hundreds and thousands of samples. So really a surrogate assay that can be run in BSL-2 that has a reporter that can be done in high throughput is necessary. And we also need such assays to rapidly screen hundreds of plasma and monoclonal antibodies for neutralizing activities. I've already shown you some data with plasma. I'll show you some data with monoclonal antibodies as well. And so what's this system? This is something that is really a strength in my lab, which is to build these types of systems and use them to study emerging virus entry. Our favorite virus, as Steve Porcelli mentioned at the beginning, is Ebola, which is a BSL-4 agent. And what we and others have done is actually incorporated the spike protein of Ebola into VSV, or vesicular stomatitis virus, which is a rhabdovirus that's a lab-adapted strain that grows really well in the lab, is really easy to work with, and actually doesn't kill anyone. And is actually this is the basis of the Ervebo Merck vaccine for Ebola that is now FDA approved. So we like to actually genetically, and there are different ways you can do this, but what we like to do is genetically engineer the VSV to replace its own glycoprotein G with that of Ebola's. And this virus also encodes GFP, and we can put other markers in there. So this is like a nice spreading virus that makes GFP positive cells, and you can do these assays in 7 to 8 hours. So that is a huge advantage of this type of approach rather than using the authentic virus. And what you're looking at here is actually a stitched together image of a single viral plaque. These are GFP positive cells. The infection started in the center, presumably with a single infection event. And you have the spreading infection that kills all these cells in the middle, and this is sort of the active ring of infection. So we can actually, I just like this picture, that's what I want to show it to you, but we can actually plaque this virus and handle it like any other kind of virus. We can select mutants, we can do lots of things with it, which makes it a very powerful system. And I'll show you some of the data that we have now using this type of approach to look at SARS-CoV-2 entry and use it as a tool for screening. So the work that I'm going to show you was initiated by Rohit Janga, who's a research assistant professor in my lab, who has really has this knack of rescuing these recombinant VSVs. He and Eva Mittler, another research assistant professor in my lab, have really shown that they can rescue some very challenging viruses in this VSV context that a lot of other folks have had difficulty with. But this virus is really hard even for Rohit. So he replaced the spike protein of VSV with that of SARS-CoV-2. Initially the virus grew very poorly, but then it started to pick up. And what he found was that it made these giant, these are not single cells, by the way, in the lower panels. These are syncytia. These are fused cells, hundreds of cells fused together, making these giant scrambled egg looking things. And just for scale, the upper panel show you GFP positive infected cells infected with a virus expressing the SARS-1 spike that Eva Mittler in my lab made. And you can see that these are much, much smaller because they're single cells. In contrast, the SARS-2 VSV makes these giant fried eggs. And that's one reason we realized that the virus is poorly infectious. But Rohit and also a whole bunch of other folks in the lab, the whole COVID crew, pitched in to serially passage this virus. And actually, what they observed was the virus started to grow better over time. And this increased growth from passage 1 through 5 through 9 was associated with first a single cell infection, but very poor, and then transition to forming these syncytia. This is passage 5. And then by passage 9, you're getting more normal looking plaques with a few syncytia, but most of the plaques don't actually have a lot of syncytia. So it seemed like the virus went through this transition where it started to produce a lot of syncytia and that allowed it to spread. But then over time, there were other adaptations that sort of allowed the virus to maintain and even accelerate growth, but do so without causing syncytia. We're still working through the molecular mechanism of this, but we have some data that I can share. So this is just showing you that individual viral plaques that were picked and amplified from a late passage all have very high titers. These are 10 to the 7th. This is a virus that started around 10 to the 3 to 10 to the 4th. So a 1,000-fold increase in infectivity as a result of serial passage and mutation acquisition. And so this is you don't need to don't worry about looking too much at the details here, but we obtained really quite a veritable zoo of different kinds of mutants. But what a lot of them had in common was a deletion of the cytoplasmic tail. So the cysteine in the C tail mutated to a stop codon that essentially terminates the protein. And we think what this does is actually retargets the protein from the ergic compartment to the cell surface. So coronaviruses normally bud into the ergic, the proteins in the spike and other membrane proteins are targeted there. And the viral nucleocapsid pore buds into the ergic. And then this sort of intact particle that's in a membrane is targeted through the secretory pathway and is released from the cell surface. Well, that's not what VSV does. VSV likes to go to the cell surface and bud there. And so what we think's happened, and we've seen this before for hantaviruses as well, we think that this deletion just removes these ER retention sequences and retarget the spike to the surface. We haven't proven that, but we think it's quite likely. Other people have seen something similar. And so this is not a normal adaptation for this virus. This is one that happens in the context of the surrogate system that we're working with. We also obtained other mutations that are very interesting because some of them are actually observed in the real virus in patients. And so we really want to try and understand which ones of these are in that category and what they might be doing, and which ones are maybe just artifacts of tissue culture passaging of coronavirus and which ones might even be specific to our VSV. So we have a lot of work to do here to deconvolute all this, but we think there's some mechanistic gems hidden in here. There are mutations we think that alter this flipping in and out of the receptor binding domain that's thought to be central to receptor binding and triggering of membrane fusion by this virus. We and others, including people studying the real virus in tissue culture, have found that the virus likes to mutate out this furin cleavage site that's between the S1 and S2 subunits. This seems to be important in vivo, but actually may be deleterious in tissue culture. And then we also see another intriguing mutation that maybe affects mutation cleavage at the C-terminal site mediated by, in some cases, by this transmembrane protease called TMPRSS2. We think that these cleavages may actually help the virus in tissue culture by preventing syncytium formation by spikes at the cell surface. But interestingly, some of this might also be true for the authentic virus. So if nothing else, we think these sort of mutants provide tools for us to really understand how the spike works, and we're pursuing that. Just very quickly, since I'm running short on time, we put this virus through its paces. We showed that this virus needs ACE2 as a receptor, just like the real virus. So this is in BHK21 cells. We don't express any ACE2. We show that we can express it nicely from its cDNA, and we get no infection on these cells unless we express ACE2 in them, and that's associated with this huge increase in infectivity. We see similar results in these types of, either if we use an antibody against the spike protein, against the ACE2 receptor, that nicely blocks infection. And we can also block infection by incubating cells with the soluble receptor binding domain that John Lai's group produced. And so this receptor binding domain presumably inhibits infection by competing with ACE2 for binding with the virus. So all of this data really points to that this VSV expressing the SARS-2 spike requires ACE2 as a receptor to bind and to enter cells. And we also show that just like the real virus, this virus needs cysteine proteases. So ammonium chloride raises endosomal pH, and it can nicely block infection, not just by VSV containing Ebola, but also VSV containing the SARS-2 spike. We see similar, although less profound results with E64, which is cysteine protease inhibitor, as well as this FYDMK, which is another cysteine protease inhibitor. We also see inhibition with the serine protease inhibitor, but only when we overexpress TMPRSS2. So basically, all of these data are really pointing to, at least in tissue culture, the virus has different ways to get cleaved by host proteases, and these are somewhat redundant. But in vivo and in airway cells, it's thought that TMPRSS2 and other related molecules are really the ones that are doing the proteolysis. So in order to do this, we actually looked at CALU3. It's still a cell line, but it is an airway-derived cell line, an epithelial cell line, and we showed that the virus can very nicely infect CALU3 cells in a manner that depends on ACE2. So we can block infection of CALU3 cells with an ACE2 antibody in a dose-dependent manner. Control IgG doesn't do anything. And similarly to the experiment I showed you with BHK21 cells, we can take A549s, which is another human airway respiratory epithelial cell line, and we can show that there's very little infection because there's no ACE2. And when we overexpress human ACE2 in these cells, now we can get very nice infection. So again, whether we're looking at sort of garden variety cell lines as well as human airway sort of origin cell lines, the virus seems to infect in a manner that depends on ACE2. We're starting to look at the protease requirements in these cells as well. But I think more importantly, we're going to be collaborating with Rick Kittis and Bridget Gompertz at UCLA to look at infections in much more sort of tissue mimetic cultures like lung organoids. And so we're quite excited to get those types of studies started with this tool. The last piece I want to mention about this virus is that it is actually a very nice virus to use for neutralization studies for antibodies. So this is looking at serum and its ability to neutralize infection. Again, 100% on the y-axis means no neutralization. So as we add more and more serum, we get this nice dose-dependent neutralization with a convalescent serum, but not a control serum. And this is the type of assay that we can run in like 7 hours and do in 96 or even 384 well plate. So we have throughput. A group of 3 people, Euge Dieterle, Denise Hasselwanter, and Rohit Janga, essentially have run all of the nude assays I showed you already and that I'm about to show you. So really, because of the assay is really amenable to throughput, and of course they work really hard, but it is really something that can be used, we think, to screen hundreds of thousands of clinical samples going forward. So an important piece of data I need to show you is that the neutralization potential of antibodies that we get with our VSV look similar to what we see with the real virus. So the VSV is on the y-axis, the real virus collaboration with our jaundice isamrid is on the x-axis. We get a very nice correlation for neutralization potency. It's not perfect, but it's good enough, we think, for screening. So we have this assay. We've shown that convalescent plasma contains these neutralizing antibodies. There are a lot of studies in progress and some data already out that convalescent plasma has value in improving outcomes in moderately to severely ill COVID patients. So, and we also know that monoclonal antibody therapies have been shown to be effective against other viral diseases. So we wondered if the same approach could be applied to COVID-19. Of course, a lot of other people wondering the same thing, but I'm going to show you some of our data on this, which has come from our consortium called Prometheus, which is a U19 NIH grant to develop human antibody therapeutics against World Health Organization blueprint pathogens. The pathogens we're going after are Crimean Congo hemorrhagic fever virus and hantavirus. That's what we're funded to do. But COVID happened, and so we pivoted to look at COVID antibodies. It's a large collaboration with many, many different institutions all over the world working together. This was our first annual meeting last fall, and you can see that we have over 50 people working together on these antibodies. And so we rely on this antibody discovery platform that takes B cells, that sorts antigen positive B cells from human convalescent donors. And then through a pipeline that's developed by our industry collaborator Adimab, can actually use a yeast platform to identify antibodies as human IgGs and actually produce them for analysis in a pretty good scale, so that hundreds of antibodies can be, in a matter of weeks, be generated and screened for activity. And so the sorting was done with the same recombinant spike protein, at least initially, although we're starting to sort with other things. We sent them some PBMCs from Einstein, from our cohort, our Bronx cohort of MMC patients, and convalescents from New Rochelle and so on. And what we can actually find that's interesting is that there is a pretty good correlation between the ELISA titer of IgG, spike-specific IgG, and the percentage of antigen-positive IG-positive B cells. Really, although the B cells are a little bit more binary, but generally we find that the higher the titers of the antibody, the higher the levels of antigen-specific B cells, which is memory B cells, which is not surprising. And so our collaborators sorted some of these samples. I'm going to skip these in the interest of time, just some characterization. And then using this high-throughput neutralization assay, we screen these antibodies, and this is a very small subset of what we've done so far. I'm just showing this as a sample. We can very quickly pull out the low bars here that are the neutralizers at a single dose, and we can then run dose curves on these to actually look more carefully at the neutralizers, and then we send these to our collaborators to look with authentic virus to confirm. And this is a sort of, we have a lot more antibodies now that we're still screening through, but our best antibody from these efforts so far is this particular molecule called 56443. You learn to remember and fall in love with these numbers. If you stare at them long enough, these horrible 5-digit names for the antibodies. But this particular antibody shown in blue here is really potent, IC50 of 20 picomolar. And in a previous campaign that was done by our collaboration that was just published in Science recently, the best antibody we identified, there was actually a cross neutralizer of SARS-1 and SARS-2, which was this molecule 55688, and it had 140 picomolar neutralization. So by looking for SARS-2 specific antibody, we can improve this quite dramatically. And this antibody compares with the best that are out there right now. And so we're actually using this as a starting point to screen for more potent neutralizing antibodies from a larger panel, to further characterize these antibodies, to optimize them. We want to obviously do the virology to really understand how they're working, but there's also a preclinical development component here to really optimize these antibodies for affinity, stability, PK, and quote unquote developability. Some antibodies are simply not good candidates to be scaled up and given in vivo as a therapeutic. We also want to identify synergistic combinations and down select to lead cocktails, and in collaboration with John Lai, build bispecific antibodies that may retain the potency of the cocktail, or it may be even better. We actually want to go after novel antibodies. Right now, most of the antibodies out there that are really potent neutralizers are binding to the receptor binding domain, and most of them, the vast majority of them, inhibit ACE2 binding. We want to go find other ones by a variety of different approaches that I'm happy to discuss, and also other discovery platforms. And we're getting ready to evaluate some of our lead candidates in animal challenge models. We're starting with wild-type Syrian hamsters. And then there are plans to initiate preclinical development through our industry partners. Okay, with that, I'll stop. I'd like to thank, this is my whole group. Actually, it's missing a couple of people. We need another group shot. We'll have to do it. I don't know how we're gonna need a really like wide angle lens to get, maybe a fisheye lens to get everyone with social distancing. I'm not sure, but this is the lab as it was in last fall. And we like to dress up. And so this is sort of us on Alternative Careers Day. We have a lot of cooks and a lot of people that want to take up the cloth. So I found that an interesting observation. Is that something that science does to people? I'm not sure. And then, of course, last but not least, like to thank our funders. And with that, I'll stop. And if there's any time remaining, I'd be happy to answer some questions.
Thank you, Kartik. Really, truly amazing accomplishments for just 3 months, right?
Give or take, yeah.
Really incredible. Thank you. So,
Thank you.
I don't know in terms of how the time is handled. I think Zoom calls can continue for whoever wants to remain. Right now, we officially have 3 minutes left. Kartik, if you open up your chat box, you'll see you already have one question. It's from Hilda Yee. So you can either read the question or you can unmute Hilda and she can ask you.
Okay. Yeah, so I'm happy to read the question out. So Hildy asked, have you looked into the so-called S1 shedding phenomenon? The short answer is that we have not, but is very much on our to-do list. We think that some of these S1 mutations in our VSV are probably mutations that either promote or inhibit shedding. A little bit, the discussion reminds me of like the early days of HIV with GP120 shedding, but we'll see how that kind of plays out. But that is an important question we need to look at. From Jacqueline Achka has a question. Have you looked at the whole range of respiratory epithelial cells in terms of infectivity of SARS-CoV-2? We have not. That is something that we would love to do. We would also, as I mentioned, love to look at organoids. Jacqueline, if you have access to those cells or want to talk more, let me know, because I think that would definitely be something. Ralph Barrick published a paper in Cell where they did this. They looked at the whole gradient of respiratory epithelial cells. It would be quite interesting to do that and compare their results to ours. Sean Whalen published a paper with the VSV where they did this in intestinal cells. So I think, as far as I know, this hasn't been published anyway or reported for the respiratory cells. So
I actually agree, Kartik. Just a quick comment. We've looked into this because we are looking at that NTB, but we've also looked for other pathogens, and there's very little on these cells. We are getting them. We are getting the cell lines that represent the different. I mean, there are 4 types of respiratory epithelial cells. I'll be in touch with you on that separately.
Okay. Yeah, let's definitely talk. Pratyush has a question. Does COVID-19 make VLPs? Yeah, it's a good question. I think I remember seeing something about that, but I didn't really dive into it. So I'm afraid I don't really know a lot about that particular strategy. Certainly, a lot of viruses can make VLPs, but I don't know specifically about that one. Pam Stanley has a question. Are you planning to look for neutralizing antibodies in people who are living with patients before and during their illness? I wasn't planning on it, but if somebody has plans and they would like us to participate and use our assays to sort of support the work, we would love to do it. Seems like it would be sort of challenging to recruit and find a cohort of those people, but I know that Amy Fox, for example, is very interested in these sort of, and a lot of folks in the diagnostic space are interested in these so-called discordant families where some people have it and others don't get it. And there's some data suggesting maybe the domestic attack rate of the virus, the probability of getting infection from one person to another is actually quite low, maybe in the 20% or something. So that's a question. But that is an important question to try and answer. Hilda, you had another question. Is the ELISA antigen protein made by Steve cleaved between S1 and S2? The answer is no, because that cleavage site has been mutated out. Alan Spiegel has a question. Can you discriminate in highest neutralizing patient sera, whether it is a single patient antibody versus a synergistic combination? That's a sort of a question that we're just starting to get into now, where we want to try and understand what is the composition of antibodies in these different sera. We have different approaches that we can employ. We have different antigens that we can use for detection. We can also do depletion of specific types of antibodies using recombinant antigens followed by neutralization assays. And something that Maddie Scharf and we have talked a lot about, which is to use sort of these sentinel antibodies. Since we have all these monoclonals, we can use them as sentinel antibodies, label them, and then look for the ability of sera to quantitatively block binding by different sort of antibodies that recognize different epitopes. That would allow us to do more fine mapping. Another approach is to make panels of mutants in the spike and do something similar. So that is something that these are all approaches that we're exploring, and we hope to learn a little bit. Our starting point is to look at these discordant patients between spike binding and neutralization to try and understand why people who make sera, like who have a response to equivalent levels of spike reactivity, have such vast differences in the capacity to neutralize viral entry. So, I mean, I think this sort of touches on this question, and we hope to try and understand to what extent individual and combination of antibodies can contribute to neutralization. So John M has a question. It looked like there's positive reactivity in the pre-COVID serum samples. Is that right? So, I guess every assay has a false positive rate. And in our case, we do have some. Well, some are like, we don't really know for sure because they're like early 2020, but we do have true pre-COVID samples, and we do have some reactivity there in a like 1 or 2%. I think there are various possible explanations. In some cases, those samples are from specific types of individuals. Maybe people that are on some kind of treatment or immunosuppressive treatment, or maybe they were pregnant, or maybe they were receiving IVIG. We haven't gone and drilled into the case histories of each of these people, but at least some of them, I think, account for this. I think if Amy Fox is here, she could probably address this more specifically. But I mean, that is, I can't completely explain why there's positive reactivity in some of the pre-COVID samples. It is something that we should do, is look at the other spikes from the other coronaviruses to see if they're off the charts on those other spikes. Because if that's the case, maybe we're just getting some cross reactivity with corona. With common coronas, we don't typically see that. So yeah. But no, that's a good question that I don't really have an answer to. Jacqueline Achker has another question. Can you comment on the Nature paper published today on low antibody titers and asymptomatics with rapidly declining neutralizing titers? Seems to fit into what we've learned thus far. Well, I mean, we're just starting to do this ourselves longitudinally in the convalescent cohort. It's very, very early, and we actually haven't run the newts yet. We're doing it right now, or soon. But already just by IgE spike ELISA, we can see that the titers are dropping. Whether that's just because of sort of resolution of plasmablasts or something. And, I think we have to look at the B and T cell responses as well, and we have to look out longer. And we would also like to look at, obviously, neutralization. So that's all in progress, and these longitudinal questions are obviously really important. I think we're going to have a lot of data on this from various sources. So. Tabani has a question. Do you plan to look at T cell exhaustion and viral loads? I'm not really a T cell immunologist, so I feel like that's, I'm happy to, I've spoken with other immunologists like Gregoire Laveau and David Fuchsbin to think about doing these kinds of experiments. I think we are interested to work together with people, to try and answer these questions, but that's not really my expertise to look at T cell exhaustion. I think there are other folks on campus who would be able to, have the experience and the knowledge to really drive those kind of studies. But again, we're happy to collaborate with whoever is interested to look at these things. There's clearly way too many questions for one lab or even a group of labs to look at. So. Okay, I don't see any more questions on the chat. And I guess since it's past 1, and Steve had to leave because he had another call. So unless there are any other questions or anything else people want to ask me, then I'm going to take the liberty of ending this seminar. And I wanted to thank once again the organizers and Dean Tomaselli for putting this together. And really, I'm looking forward to all of the talks that are to come down the road on COVID from folks at Einstein and Montefiore. Thanks.